Hello everyone, welcome to the talk of talk series. So last time we heard about Daryl and maybe Daryl.
Thank you. Yes, I trust you can all see the slides. Yeah, okay. Glad to be back again. Last week we had discussed about domains and domain swapping, especially how domain swapping can be of predictive value because it's implicated in neurodegenerative diseases and so on. But at that time, the phenomenon was mostly about the exchange of subunits, right? But in today's talk, we are going to initially talk about a class of domains which are disulphide rich. For instance, in this protein example, let's concentrate on the chain in yellow. And you can see that there are actually not one or two, but three disulphide bonds. These are shown in bright yellow color, and they connect to cysteine residues, and they form a disulphide bond, which is made up of two sulfur atoms getting covalently bonded. So what is so special about the disulphide-rich, namely the covalent crosslinks? They are much more stable being held that way. As a result, they have very little chance or requirement to have their backbone structure so rigid. For instance, they do not have to have too many secondary structures, and they may not need to have a strong hydrophobic core. In fact, disulphide-rich systems lack a strong hydrophobic core, and plus the fact that they have several repeating cysteine residues means spuriously in sequence searches, it can get aligned to some other protein, which has nothing to do with these sort of systems. By the way, this chain that is shown in yellow color is nothing but bovine trypsin inhibitor, or BPTI in short. And what we had seen is actually the complex crystal structure along with the trypsin itself. However, coming to the point, these sort of domains are special because they are lacking hydrophobic core, and they can also get spuriously aligned with other sorts of disulphide systems. So they have to handle much more carefully. And with this in mind, we had started to apply this algorithm, which I developed many years ago. In fact, this algorithm was written by me as a PhD student in Professor Balaram's lab. And this has been referred as motive of modeling in disulphide bonds in proteins, where primarily the algorithm invests on the property that even though each of these cross-link bonds, covalent bonds, and that's the sulfur-sulfur bond we were talking about, even though each of these covalent bonds are single bonds, that means they have freedom of rotation. You will see that the sulfur being a weak nucleophile and has all sorts of restrictions. And therefore, you will see that the distance between the two sulfur atoms, which we will call as the matrices, and then the torsion angles that is subtended by these two sulfurs involving C beta, C beta, which is called as chiases, have very strong requirements to be in and around ideal values. The length would have been 2 angstroms, the chiases would be plus or minus 90 degrees, and so on. Not only that, you will find that the chi1i torsion angles are usually around this gauche plus gauche minus transformation. And therefore, we performed the data analysis years ago in collaboration with Professor Ramakrishnan's group and his student, late Professor Srinivasan. And we had established certain conditions just by looking at data, and this is early bioinformatics analysis, by the way. And then we noticed that RSS and chiases are pretty strict, and they're usually, even the protein observations, they don't wander around very much from these ideal values. And the chi1i, chi2, chi1j also has to be somewhat rigid. And since this is the case, if we want to model disulphide bonds into a protein whose structure is known, and we don't want to, mess around with the protein structure and make it more strained, etc., there are only a few ways by which you can introduce these disulphide bonds. And then we set about to grade them. If the RSS, chiases, chi1i, chi1j are within the limits, then it would get the highest grade of A. And if some of the deviations start to set in at the chi1i, chi1j, then it would be a grade B, etc. So the offshoot of this sort of an analysis and an algorithm is that given a protein of known three-dimensional structure, now we can begin to imagine and recognize pairs of residues where if you change it to the cysteine residue and a disulphide bond can form strainlessly. That is the whole purpose. And that means we are able to rapidly populate and if we imagine a disulphide database, for instance, the BPGI examples, where there are 3 disulphides, which we call as native, because they are truly just there in the protein. Using this sort of a MODIP algorithm, we are able to select or identify 8 others where facile modeling of a disulfide is possible. The stereochemistry permits it, the stereochemistry at the backbone permits it, and also at the side chain covalent crosslink. So this allows us to rapidly expand the disulfide database, then not only into including the native disulfide bonds, but all those which are permissible stereochemically. And that means we are able to escalate and collect a huge amount of information, which simply suggests that these are all the substructural fragments in various protein structures, wherein the introduction of disulfides could have no stability effect or so, right? And this is how the database of disulfide bonds came into picture. And at that time, when we escalated it as a database, there were 2 people, and of course, the MODIP was a key player and the heart of the development of the database. But the people who worked on it were Vinayakam, and then Rajesh, who particularly then applied it on disulfide-rich systems with the purpose of modeling in mind. And when we submitted it to the journal and nucleic acid research, and there are special database or web server issues where you can project these sort of useful repositories. And when we submitted it to NAR, in short, what we received is one of very interesting comments from the reviewers. And they said, we had started off with a non-redundant, that is, not the same sort of protein coming again and again. And then we had developed this database. And then one of the reviewers said, why don't you apply it on the entire PDB? And then we were a little flabbergasted. I mean, it's going to be a lot of work. And then what if there is redundancy felt at the level of the database? But it turned out that it was useful because depending on the conformation of the protein, even though the sequence may be the same, there seems to be a variety where these sorts of sites where disulfides can be introduced can become recognized. And Vinayakam is one person who is ever smiling, and he starts to smile even more if there is more work to do. And he was simultaneously handling a few other databases, but there we were. We populated the database to look at the entire PDB, and then the first version of the DSD base came into picture. Right, this is still we are part of the first story. And like you can imagine, the first version of this DSD base was in the year 2004, and a few years before that, because we started work on it at least a year ago, right? But then we had an intermediate version sometime in 2014, when Soumya Indrakumar, who is an integrated PhD student in Indian Institute of Science, had a primary guide who was Srinivasan, and together with Murugavel had tried their level best to update the database by means of populating newer entries. However, this part of the work did not get reported in the form of a publication. But we have Neha Kalmankar, who kind of revived and re-examined this problem in the context of her PhD project, which was on a disulfide-rich set of systems, none other than the cyclotides that are present in some plants. All right, in this context, Neha Kalmankar had actually then repopulated the database, and it is now also not only publicly available, but we have a short report that kind of mentions just this, right? So this is how the first story goes, and Neha is here, even though she's in Oxford, she's here with us virtually, and she's going to describe her point of view about how the population of this database became possible and her experiences, right? So Neha, please.
Hello, everyone. Am I visible and audible?
Yes.
Hello, everyone. Happy to be here today talking about a part of my PhD work. I'm Neha, and I'm currently a postdoctoral research associate at the University of Oxford. And today I will talk about a work we did during my PhD, which involved updating a database called DSD base. And as, sorry, I think, yeah. As ma'am introduced this database already, it is a database of disulphide bonds in proteins and gives information of not only the native disulphides that are present in proteins, but also that are stereochemically possible between pairs of residues in a given protein. And the first version of the database was created sometime in early 2004 or in 2003 by Vinayagam and group. And at that time, the database included over 19,000 structures from the protein data bank. And that was the April 2003 release. And you can see that as the protein data bank, the number of structures deposited in the protein data bank grew enormously over the years, over the decade. The database also had to be updated to include all these disulfide-rich protein structures into the DST base. So we went from 19,000 structures to close to 1,53,000 structures, taking into account the January 2021 release of the PDB. And this was a substantial increase from the previous database, but because it was nearly 17 years or so, even though we had intermittently also updated the database, and that helped enormously for me to make a comprehensive inclusion of all the PDB entries. But it was still quite a large number of entries that had to be included into the database. And it was a fun and exciting time because I had never done such a work before. And this slide actually shows the statistical comparison of what the database version 1 looked like and what the latest database, which was released in Jan 2021 database looks like. And you can see that with the growing structures of PDB, even the DST base database has also grown enormously. But this is how the DST base web server, the latest version, the browser looks like. And I will take you through some of the different resources available and tools that have been included in this database. The users can now search this database using keyword search, either by inputting a PDB ID or PDB title or like a EC number, and can browse through all the different entries. But there are also several tools that have been made available. One that is MODIP, which ma'am has introduced, and a few other tools like RANMod and modeling peptides, and several other features and interface resources have been made available in this database. So one of the tools, that is MODIP, is a very important tool. It's parallel to what one does in experimental work, where they do site-directed mutagenesis, where they want to include disulfide bridges between pairs of residues to increase the stability of a protein. And this, instead of doing experimentally, can be done in silico. That is, you introduce in a strainless manner cysteine residues at 2 positions, at a pair of residues, and the MODIP algorithm or the MODIP program now creates a disulfide bond between these 2 pairs of residues. So this is the output page of this MODIP program, but the user is basically required to input a PDB structure, and it searches for all the possible sites along the polypeptide backbone where strainlessly a disulfide bond can be introduced. And in this particular example, this is an example of a xylanase protein, which has, and in this structure, you can see these are the different positions where disulfide bonds can be introduced. But this particular position, serine 110 and asparagine 154, at this position, the disulfide bond that can be introduced is of the highest quality grade, grade A. And the user can now download this PDB structure and view the stereochemical parameters of the disulphide bond that has been introduced here. The stereochemical parameters include the SS bond distances or the disulphide angles of these disulphide, the mutated disulphide bond, and the different grades. So this is what the output looks like for the MODIP program. The second tool is RANMOD, which is a program for random conformation generation for a polypeptide backbone. And this program is an ab initio way of modeling three-dimensional structures of disulphide-rich polypeptides for which we don't have structural information already present, and we know its sequence and we know the disulphide connectivity, but we don't have any structural information. And this is not like a homology-based modeling method. It's basically the program generates and assigns permissible backbone conformations for a given polypeptide sequence at every residue position, and it searches the conformational space for all the models that can incorporate these backbone but allowed conformations at every residue position. And the MODIP algorithm, which is built into this RANMOD, creates the disulphide bond between pairs of cysteine residues. And the user now can input the sequence in this text in this box and has to provide the disulphide connections. But if you also have secondary structure information of the given protein, you can input that as well. And then the program searches the conformation space for all the possible, permissible, and allowed conformations for that given polypeptide. So this is an example. This is a conotoxin sequence, which has two disulphide bridges, and this is the RANMOD generated model, and you can see that the RANMOD model has a close resemblance or close similarity with the original NMR model of the same disulphide-rich polypeptide. So it's in well agreement with the NMR structure. And the RANMOD program performs really well for even higher, for case studies which has more number of disulphide bridges, like 3 disulphide bridges as well. The third tool that I will talk about briefly is about modeling polypeptide. It's called Modeling Polypeptides tool. And this is where the user can probe the database and search the non-redundant data sets. There are different non-redundant data sets built into the DST base. And it can, the user can probe this database with a particular cysteine or disulphide bond connectivity, and it searches for close homologs or similar protein structures or peptide structures or even substructures of a given polypeptide for the same cysteine pattern or the same disulphide pattern, and the user can input the sequence information. This is optional. You don't need to put the sequence information. You have to put the disulphide bond connectivity that you are interested in, and it searches through the database for the same pattern. And this is an example. This is a disulphide, 3 disulphide bonded connectivity, and it searched through the database. This is the connectivity that I input, and it found a close homologous structure of this particular connectivity. And I think, if I remember correctly, it bears about 75% sequence identity with this particular hit that was generated. So to summarize, the DST base version 2.0 now includes over 1,53,000 PDB entries from the Jan 2021 release, and it has over 2,16,000 native disulphide bond segments, but also over 20 lakh model disulphide bond segments from the PDB Jan 2021 release that is included into the latest version. Now you can model the disulphide crosslinks using the MODIP algorithm. and the user can search for multiple disulphide bond containing loop segments within from the database, but also now you can annotate these substructures or segments for their function using geoterms and even subcellular localization. And the difference between the previous DST base and the latest DST base, though the previous one also had MODIP and you could search for the disulphide bond segments, but the tool to model the three-dimensional models of disulphide-rich polypeptides was not available previously, but has been now included into this DST base, and that is the RANMOD program. Now you can model the three-dimensional models for disulphide-rich peptides for which you don't have any structural information, but you know the disulphide connectivity. You can model these structures. So that has been included, and the database can be accessed using this following link. And these are some of the references that are relevant to this work. So thank you. If any questions are there, I can answer them in the Q&A box or even now. Thanks.
Thank you, Neha. Thanks for explaining the disulphide database part of the story so well. And we now would proceed to look at the second story. We have 3 more to go, so stay on. So the second story is also an old one. It predates the formation of the lab. It's a part of my postdoctoral interest, in fact, which is to look at domains that are remotely related in sequence, but they have remarkably similar structures, and one example is shown up here in the slide. And what is also useful to appreciate is that not only these protein domains are distantly related in sequence, but are remarkably similar, they have 2 other aspects to note. One is that their biological functions are rather similar. They are not exactly the same. In the enzyme classification, they won't fall into the same 4-digit category, but they are rather similar. And they also have varying amount of extra residues, which could themselves be secondary structures. So this becomes very important to appreciate that computing methods are required to handle such cases, not only because their sequence identity is low, but they have large number of what are called as indels, insertions and deletions, which have to be handled appropriately, because in the process of arriving at a sound, pretty looking alignment, we do not want the wrong helices to be under each other, or what we call as equivalent residues. And for this, we resort to a program called Comparer. And this was written by none other than Andrej Sully when he was in Tom Blundell's group. And this Comparer program stops to worry about amino acid identities or even similarities, and instead starts to look for structural similarities, namely secondary structures, the hydrogen bonding pattern, and these are driven to obtain a nice looking multiple sequence alignment of the members that we believe belong to that category, what we call as a superfamily. And in doing so, you can imagine that the equivalent residues are placed such that they have structural and biological meaning. So this was an attempt made when I was a postdoctoral fellow, wherein we had many computing people in the lab who volunteered, and each one of us used to adopt a superfamily and start to work on it. And this was all, once we are happy with the evolutionary model, we can use it for all sorts of derived analysis. And one example is the cusp that already Sandhya had explained a few weeks ago, wherein we look at the conserved and unconserved blocks, right? And this repository first version got published in 98, and it was called as Campus, Cambridge something, protein alignment, structural superfamilies, etc. So, but when I moved over to Bangalore, we had an understanding with my postdoctoral mentor, Tom Blundell, that I will carry this project with me. And it's so beautiful journey, what I have tried to project here in this slide for you, which is to start off looking at the campus itself, which was formed in 98. And as soon as the lab got established in Bangalore in NCPS, we managed to develop the next version of the database, which we now call it as PAS2. And then this was published in NAR, Nucleic Acid Research, and it was a very satisfying sort of achievement, I must say, because we started from scratch and then were able to develop the first PAS2 version. But then 3 years ago, we had a second version, and then the PAS 2.3 is in year 2008. And you can see the journey is so beautiful, it is continuing. And the players are different, but not totally different. You will see that there is a insufficient amount of hand-holding. At least one member is common between two consecutive updates. Not only that, this enabled the transfer of technology, as it were. Because not only we are using comparer, we have to supply some clues as which are equivalent regions. And for that, we had a lot of opportunities. And not only we were going to handle more and more entries, and the database is growing with time, definitely. But as though we are going on a window shopping, newer and newer softwares were getting available. And we had to make a choice every time. Which software are we going to use before we put it into the comparer routine? Plus, we were always able to appreciate higher amount of difficulties in containing members that are already rebelling in their structures, right? And forming not only loop regions, but also would not easily superpose, and we call them as outliers. And we were also able to offer functionally information such as functionally important residues and so on. So this journey has been a very beautiful and continuous one. And I have requested Gandhimati, who's going to share some of her experiences, and she's somewhere in the middle. And definitely, like many others, she is in 2 updates. And she would, for instance, explain her experience looking at the outliers and how we had tried to understand them. We will go hear the recorded details, the audio recorded details that come from Gandhimati's point of view. So this is the second part of the story. Please stay tuned.
Hello, everyone. I am Dr. Gandhimati. I completed my PhD in 2014 under the supervision of Professor Saudamani. And I'm happy and excited to be part of this CAPS talk. And I'm also feel proud that I'm also, part of this 25 years of CAPS journey. I would like to thank Professor Saudamani for giving me this opportunity to present to the team. Thank you. So today I will be talking about PAS2 database, which is a database for structure-based sequence alignment of distantly related SCOP domain superfamilies. So as you all know that homologous protein sequences can be easily grouped by simple sequence search methods. But distantly related proteins, which means proteins related at superfamily level, they are characterized by poor sequence identity, but they have a very good conservation at the structural level. So these distantly related proteins can be grouped only by structure-based sequence alignment methods. So why do we study distantly related proteins? So one reason could be we can understand structure-function relationship at superfamily level. And the second reason, like if someone is modeling distantly related protein, these structural information will be really useful to model the protein. And then with this introduction, I would like to discuss more about PAS2 protocol. So this is a protocol we followed, and this protocol is published in 2012 in Nucleic Acid Research. For this, we have used SCOP 1.75 version with the 1961 superfamilies. So when we define how we define superfamilies, so all those protein domains which are less than 40% sequence identity, they have been retrieved and they have been classified as 2-member and multi-member superfamilies. So these superfamilies needs to undergo structure-based sequence alignment. So there are 2 phases of alignment. One is the initial and the final alignment phase. So in the initial alignment phase, after careful assessment of structural alignment tools, we observed that MinRMS MAT can do efficient structure-based alignment compared to other tools. So we have used these tools for initial alignment. And the final alignment phase was done using comparer. So comparer is a very efficient program which was developed in 1990 by Professor Tom Blundell and his team. And this program uses a lot of structural features to identify a reliable alignment. So comparer requires certain initial information on the alignment. That means it requires certain guidance, meaning initial equivalences, like structurally there are multiple domains, what kind of a structural similarity they have and how they have been aligned. So a rough alignment can be done by any tool, and then comparer program was used to refine that alignments to make it more, efficient way of aligning those distantly related proteins. So once we have aligned those superfamily level alignments, then we have done certain assessment, assessing the alignment, the superfamily level alignment. One is that by checking in their mean RMSD. So if the alignment is really good at superfamily level, they would have less than 5.5 Armstrong RMSD. And they also have a good secondary structure equivalences. Minimum of 30% of secondary structures should get aligned in a superfamily. So these two are important alignment assessment phase. So once we assess these alignments, then we take them for further structural annotation of these alignments, such as looking into their structural motif and also deriving HMM profile for the same, and also bringing out the alignment statistics, what percentage of sequence got aligned, structural elements got aligned, and phylogeny, how is the relationship of these protein domains at superfamily level structurally. So all these informations we derive, which are useful for studying the superfamily level relationship. And the members which they have more than 5.5 Armstrong RMSD, we call them as outliers, and we try to exclude them, and the remaining members are considered under superfamily. So we also looked into outliers a bit more closely. I will talk about it in the next few slides. Next is about PAS2 statistics. As I told you, PAS2 database has a very long history, and it is actually initiated by Professor Saudamani in 1998 in the name of campus. And we have subsequent versions. I mean, every time there is a SCOP update, and we have a corresponding PAS2 update. And now last year we are done with PAS 2.7. And I was also fortunate to be part of two updates, PAS 2.4 and 5. And we have introduced, every version has challenges, and we have to rectify those challenges. And there are new features got added in every version over time. And yeah, this is something I would like to emphasize. And I'm also proud to show this slide with the different, multiple versions of PAS2. What are the improvements we have done in the recent version? So from 2004 to now, we have recently we have added few additional features to the database. One is assigning new structural entries to pre-existing superfamilies. So we have generated HMM profile from the alignment. So using those HMM profile, we searched in PDB to gather more and more structures, which could be which could be part of that superfamily of interest. Second, mapping gene ontology terms, definitely mapping those Go terms to identify probable functions, which is useful to understand the superfamily more detailedly. Then the third important one is mapping fully conserved residues. So when we were doing this, we found that there are certain residues which are 100% aligned across all the domains of a superfamily. The superfamily could be 2 domains or 40 or 50 multi-domain, multi-membered superfamilies. Still, we identified certain residues which are conserved across complete all the members. So that means they are at superfamily level, they are conserved. So that shows very interesting information that they could be functionally important residues, and they might play a they might contribute at the functional level, even at superfamily level. So we started mapping them, reporting them. So these 3 are the important additions to our recent versions. So as I mentioned, I would like to talk more about outliers. So previous versions, we used to see a lot of outliers, but it was not heavily focused on outliers. Then we started looking into them, like, what are the reasons for these outliers, why they are part of the superfamily, but they are not been, having a proper structural or highly similar conserved structural core. So when we see them, there were superfamilies with single domain or single protein domain reported as outlier. And there are superfamilies with multiple members, more than one as well. So we started characterizing them. We found the reason could be either they are structural different, that means just merely a structural, topological or structural difference happening, and no other, reason behind it, or they could be functional. Definitely structural, functional can be interchangeable at the outlier level. Then we have also seen certain multiple outliers having a similar kind of a structure. They form subgroups. So the first example is all the three domains belong to a family, but there is one structure which is slightly, having a different topology compared to other two domains in that superfamily. And the second category is they are functionally different, which means the domain is an outlier, reported as outlier, but it belongs to a different family. So that means they're functionally different as well. And the third category, wherein we see multiple outliers, but they have a very good structural similarity among them, so which can get clustered among themselves. So with all these three kinds of types we have observed, we started characterizing them in the past 2.4 version as multi-membered, from the multi-membered superfamily. We checked for structurally deviant members. We identified around 159. We started categorizing them. Are they family-specific outlier or just only structural outlier? Are they also forming some subgroups and all? So this complete work has been published in PLOS One in 2013. And this, we have named it also as ribbling for reason. This was given by Professor Saudamani, the title of this paper. Reasons. So I'm just going to give some examples of these outliers. So why there are, what kind of a reasons, right? Okay, we know structural and functional, it could be functional reason or subgroups, but what kind of reasons, structural or functional reason, why they become an outlier? So we have seen, say, certain case studies, certain interesting examples. One is different in secondary structure composition, architecture, and topology. So you could see this ADC-like superfamily wherein all the members follow a similar topology, but just one particular family of members have a different secondary structural architecture and topology, which made them to be an outlier, but they all belong to the same superfamily, but they are functionally, they are different. That means they belong to a family. The second example, duplication in secondary structure. We have seen all the members having a similar topology, but there is one particular domain have a duplication of the secondary structures. So that is also one interesting example. The third is circular permutation. And in this superfamily, every member or every domain have a similar secondary structure topology, but one particular member have a circular permuted topology, which also comes with the specific function. So this is FAD linked oxidoreductase superfamily. I have more examples. The next one is domain swapping. Domain swapping is an interesting phenomena wherein the secondary structure, equal exchange of secondary or parts of secondary structures across dimers or oligomers. So we have also observed one example in this category. It's a polar box domain wherein one domain, a part of secondary structure has been exchanged across dimers. The next is diverse elaborations to the core. So as you can see here, there are multiple members in this superfamily, but one domain which had an additional elaborations in their C terminal and N terminal, which goes with the function. That means it is involved in membrane fusion. So that's why they have this elongated structure. So this is also one interesting example we have analyzed. And the next category is on change in EC number. Obviously, when there is a different EC number and they have different enzymatic activity, there's an alpha helical pyridoxine superfamily. So all these examples are detailedly explained in the PLOS One paper. And this has given us, this has given us a lot of idea about, okay, we cannot leave outliers. It's like that. We need to start looking into them. If there are outliers, how we can, group them within outliers if there is a subgroup possible. So from the next versions onwards, we started looking into these outliers, trying to add them as a superfamily kind of a category. So that's how we started, understanding these outliers from past 2 version onwards. So summary of my talk. So we identified match followed by comparer works well for most of the superfamilies, and comparer can efficiently recognize outliers present in the superfamily. And these structural deviant members literally, demand for structural function paradigm to be re-examined. And also we have to look for reliable structural classification programs. So with these summary, I actually past to have given me 4 publications. One is on the outlier, another one is comparing the multiple structural alignment programs and identifying which is the most efficient program to align distantly related proteins. And I was also part of 2 past 2 updates. And I would like to thank Professor Saudamani and all the co-authors for giving me the support. And of course, I would like to thank all the CAPS team members for all the support and help. Thank you.
Thank you. Yeah, even though it is a recorded one, I wish to thank Gandhimati for explaining her part of the journey in the second story. And we also have Woman Matthew here with us. And Woman has been the key person who will put data in for use. For instance, you can find his name in multiple updates, like last 3 updates, and he's going to be also there in the current one here. So thanks, Woman, for coming online, and please do stay on because then we might have some questions. And that was our second story. And also I wish to thank GA for kind of introducing the point that functional residues are usually 100% conserved. And wherever there are, the other way around of looking at it is if there is 100% residue conservation, perhaps we can, at the superfamily level, perhaps we can begin to attribute a functional property to such alignment positions. But it's as though nature can become quite naughty. And sometimes you can have variations which go all the way even at functional residues. And therefore, you may find, let us say there is a classic motif, a spatial motif such as a triad for enzymes. And such residues may not be observed in the newly recognized family members. So does that mean that that particular new family member should not be belonging to that superfamily? Does it mean that it is an inactive member of that superfamily? So there are many questions that arise. So this, what I have tried to do is to pick up some statements from this paper that I very much enjoyed reading. So this is a paper again by Janet Thornton, Professor Janet Thornton's group back in 1999. So for us and for most scientists, reading papers have different purposes and you can reread papers for different reasons. And many times you can get inspired and come up with ideas and also to strengthen your own ideas. For instance, here they have said that the information is not always available electronically for function inscription. And function itself is a very hierarchical term. And one might then need to accommodate this sort of distinctions by means of careful structural analysis of domains. And it also goes on to say at a different point that automatic tools that search for structural or functional motifs need to be considered, and they should not actually be constrained by the amino acid order. That means you can, if the functional motif and those residues are missing, perhaps you should look like, you're working on a compass. You should just sit at one of the active site residues and look around for alternate residues that may be playing the role. And this actually kind of inspired us to code algorithm by which you can look for residue preferences given a protein of known structure. And I can tell you very, very, very quickly how this became useful. And are you able to see the slides? Yes? Okay, because I'm just trying to go to the next slide. Are you able to see the slides? No?
We see the same slide, ma'am.
Ah. Okay, I'll do this again. Okay, so you see the pass through trajectory where you can see it's woman's name multiple times that we were talking about. And then let me go quickly to the slide. Yeah, this is the slide I wanted to pick up, where we recognize by reading this paper by Professor Janet Thornton's group on the requirement to be more flexible to in searching for functionally important residues where changes can happen. All right. And this particular next slide shows you where our role became relevant and why our lab, CAPS 25, CAPS lab had started to write this algorithm. And this is also what is called as what I refer as a birth of Hori. Because we had a visitor called Mike Merrick, who had actually come to visit Professor Matthew's lab. And he's from John Innes Center in the UK. And as a visitor, he was chatting with me and over lunch. And then he talked about a soil bacteria, which seems to be producing a large amount of an enzyme for which he did not know the structure nor its function. So that's where we started looking into the sequence, and we had very high confidence that it belongs to the superfamily of alpha beta hydrolases, but the functionally important residues were not all in place. So when Anirban Badri, who has helped us with this sequence analysis, examined and could not find the active site residues, I started writing this program rather in a very naive manner. And you can see the date is actually, it's dated 2007. But if you look into the comment column, I have already actually started writing in 2001. And what I was trying to do is to sitting on one of the active site residues. If the other two active site residues are not in place, can I go around searching within the protein structure and look for a compatible other partner where we hypothesize that all of them will participate and perform the functional role. And this kind of led into this sort of a beautiful server, which is called as Hori. And really speaking, we had a very talented intern from Anna University called Rashi Srinivasan, who joined the lab. And together with Sundaramurthy, whose PhD thesis was going to be on the sort of residue interactions, and geared by the help of people like Shami in the lab, we then created this web server called Hori. As the name suggests, it's going to help users to look for higher order residue interactions. They're not limited by residue pairs. We can just look around for clusters of arrangements. For instance, you can use this to just take a bunch of residues that you're looking for, like the alpha beta hydrolase example, and then search all around the protein. Or you can do, you can ask this server to report all possible interactions, and it's a Hori global. And then you can also do something which is a residue based and say, please give me all the serines, aspartates, and histidines within certain distance. And the server is going to report it back. So this was a very nice sort of an exercise. And I mean to say that it has gone through several changes. And now the next few minutes, the current student, one of our current students, Sarthak Joshi, is going to explain how he has been reviving this algorithm from his own viewpoint. So I call upon Sarthak to present Hori in the current version.
Thank you for the introduction. So today I will be showing the Hori web server and my version of it. So before I go into the Hori web server, I would like to first talk a little bit about 440 units level propensities, and this is why we stopped at quadruple interactions and did not go wrong. So we assume that all amino acids are kind of equally sized spheres, and if that is the case, we cannot have more than 4 spheres mutually interacting with each other, which leads quadruple interaction being the highest level of higher order interactions possible. So CAPS web server had managed this slide. Looks like this right now. It has 3 modes: global, like where global mode does the whole protein, the light mode within set with residue for. In cluster mode, you can have interactions of specific pairwise interactions between the body interactions. I will show you my version as now studying. So before we go to my version, I would like to tell you the different uses of coding. As ma'am said, ma'am described a very specific use case of coding, where she wanted to investigate one specific protein and if the active site residues are all in place or not. As we know right now, a lot of AI tools require a lot of data, big data sets. And using coding, we can generate large data sets which contain residue interactions of multiple proteins. And then using those data sets, predict various things, like you can predict the folds, you can model proteins, and you can also look at protein-protein interactions using data. What my version looks like, it is a command line tool which as you can see, has a lot of options. You can have interchain or interactions. You can also view the interactions on Bioinformatics. You can show you another. You can select if you want the center of the residue to be C alpha or C beta. You can also select if you want a specific chain or do you want interactions with all the chains. So I think
Is the voice clear? Someone has chatted in the box saying the voice is not so clear. We'll see why. Maybe yeah.
better.
Yeah.
So okay, I'll show you a little demo of one protein for on your terminal. Just write query.py. You can. This is an interactive mode where you can tell it you want to have interchain or interaction. Here as a window. With a PDB ID, here you can also give it a file name. If you give a particular file name, it will look for that file. If the file name doesn't exist, you will download the PDB for you. Download the PDB. You can enter the distance cutoff for the interactions that you want, and it finds the interactions. It prints it on a screen. It also gives an output file, which is easy to analyze. And once it has calculated the interactions, it will ask you if you want to view the interactions or not. If you say yes, it will show you the interactions on Bioinformatics. Here, the yellow interactions are pairwise interactions, the blue interactions are triplet interactions, and there are no quadruplet interactions in this particular case. So you will be able to see it. But yeah, so here you can see all the pairwise and the quadruplet interactions on the protein that we just input. I will show you one more demo where we don't see the interactions. We also don't use the interactive mode. So in this case, we can just give it an input residue. It will output interactions in a file. And if we use it this way, we can integrate it in our own scripts where if you want to find interactions from multiple proteins, you can just give it an input to and I mentioned the dash flag just so that you can see what is happening on the screen, but that is not. So you can see it very quickly finds all the interactions in this protein. It's a very small protein. And this is how the output file will look. It is a comma separated file where it will give you the type of the interaction in pairwise, quadruplet, and triplet, and it will give you the residue number, type, and chain. It is chain residue. And this output file you can further use to analyze what okay.
Thank you, Sarthak, for explaining what goes at the back end. So we got to also hear how things work when we develop algorithms. Okay. I also want to now very briefly present the last story. Sarthak explaining that the Hori has applications which extend more than just looking for residue or preferences, or even higher order residue interactions sets the stage for us to discuss the last story, which is on what I call as harmony. And harmony is an algorithm that, again, Srinivasan and myself had done as a weekend project during our postdoctoral tenure, but it kind of acquired its own structure and became a web server after the CAPs came into picture. And the higher order residue interactions, as Sarthak mentioned, has its applications that go far beyond understanding of functional residues. it can be a central core for other bioinformatics tools such as protein structure prediction, protein modeling, and also being able to validate given proteins whose three-dimensional model we have in front. Because fortunately or unfortunately, most protein modeling algorithms will simply accept an alignment and the template from the user. So if the user become either naughty or become unaware that it is the wrong template, the model algorithm is just going to present the 3D model. Unfortunately, such models are very hard to recognize as being wrong ones, because once it goes through typical energy minimization, the energy of such models will look right. But there are things that can help us to recognize, such as residue interactions, as built into Cori and described in the previous story, which will enable us to know which is the correct fold and which is the misfold. And here is an example where we had purposely taken an immunoglobulin sequence, which is threaded onto the right structure on the left, and it is threaded onto the wrong structure, as shown on the right-hand side. And the wrong structure is a completely different structural class. It's made rich in helices, whereas we know immunoglobulins are rich in beta strands and beta sheets, right? So surprisingly, the 3D model after this misfold and wrong threading looks right from the energy point of view. However, you can imagine, just like how in a classroom, if you put in the wrong set of students in the wrong classroom, for instance, a science batch student, if you put it in a social science or a art class, they are not going to be happy. So that is one way of recognizing when we encounter wrong models, which is individual amino acids. And if you look at their local environment, are they happy? And for this, just like how we have early Chow-Fasman propensities to understand the rules, the guidelines, and the preferences, here also we look at large number of protein families and larger number of protein structures that constitute the families, and then begin to attribute propensities or probabilities in this case as to not only which amino acids are present in certain secondary structures, but other features such as hydrogen bonding and solvent exposure. So as a result, you will create what is called as an environment that is made up of 3 characters, such as H1Y. In this example, it will stand for an amino acid which has been brought into an environment where it is in the middle of a helix, it's kept solvent buried and involved in hydrogen bonding. So the question is that amino acid happy or not? And for that, we have to consult a large number of substitution tables. And you can have 30 environments if you have so many variations in secondary structures, or you can have 216 environments if you had a finer grids in the Ramachandran map to define the backbone conformation, or you can have 72 environments and so on. So depending on the number of environments, you will have those many substitution tables that are amino acid substitutions. And then you can begin to ascribe score by which you will imagine higher the score, the more comfortable that particular residue is. So if your ascribed fold is right, all the scores must be positive, high, and the total score must be a high number. However, if you compare it with the random score or a reverse amino acid sequence, if they start to clash, this means there could be local errors that are present. And those regions which acquire poor harmony scores could be recognized by mapping on the structure, where basically gray color means good, and orange or yellow or red means we are into some problematic regions, and that part of the protein has not been modeled very well. All right, so this is how the harmony works, and it has been widely used, I understand, by many protein crystallographers in the structural biology community. So we're happy about it. Okay, so these are the 4 stories I had to present, and I will very quickly take you through the people and where they are. Rajesh, who came for a 2-week training, started to accept this as his PhD problem and participated in the disulphide database. Vinayakam, as I said, now he's a director in the Takeda Pharmaceuticals in the US. Neha, who presented the DSD base update, she's currently a postdoctoral fellow in Carl Robinson's lab in the Oxford, UK. And we have Murugavel, who is currently a freelance bioinformatician And the Soumya is currently somewhere in the Netherlands in a digital software digital industry after completing her PhD. All right, in the second story, just like the trajectory showed, many players have been there. So what we have tried to put out in this slide is a collage of the people who have been in different updates and where they are right now. All right, and some of them are currently with us. And you can see Gandhimati is here, Elucida Corporation, and Uman is here. He is an associate director in the MedGenome as we speak. And you may be amazed to know that none of these people were ever recruited to work in the PAS2 project. All of these are volunteers, and every one of them have their own other projects, their main projects, and they would always very enthusiastically come forward. And I've been continuously so fortunate to not only be able to continue this project and do the updates, but to have some fine set of people. And this journey has been so exciting, and we are still learning. And in the third story, I talked about on the higher order residue interactions. This was started, as I said, by Rashi, who worked with the PhD student Sundaramurthy, who was a joint student with Gakar in IIT Roorkee. And then we have now Sarthak, who's continuing the project. And the fourth story, which I talked about harmony, this was started off by myself and Srinivasan, and it was brought in as a beautiful server by both Pugalendi and Shami. And there is some common theme in all of these four stories, because I happen to have written the algorithms for all four projects. Not only that, I always been able to go through the joy of naming servers, naming our algorithms, our modules, and then giving the title for the paper and debating about all this. These are all been very beautiful. So with this, I will now stop and request actually Shafi to take you through the list of publications. But before that, let me just show you a glimpse of the history, some random things. And this is one algorithm that I had written in 2001, which was the birth of Hori, like I said. And look to the right and please focus on what is being captured as an email. I hope you can see it still. Yes? No? It has gone? Okay, I'll try to project. There we are again. So what we can see on the right-hand side is a snapshot from my inbox. And I have a habit of retaining emails. And this is an email which I was very intrigued because this is dated 26 November 2014. And it says, it is from Gandhimati, and I call her as GA, all right? So she says, "Dear ma'am, I have been coughing too much, and it might be quite disturbing for others. Can I do work from home today?" And the subject of the email says work from home. And please remember, audience, that this is in 2014, much before the COVID lockdown ever began. And just to show you that this has been successful, there is GA sending another email saying, "I would like to inform you that the user problem is still not solved, but we have been able to solve something else." Okay, work from home works. And it had worked in 2014 itself. Okay, thank you. Shafi, please can you project the list of publications and also announce the title for the next talk. Thanks.
Yeah. Hello, everyone. So these are the most cited research from today's talk. First, we have disulphide rich section, and we have 3 early papers from MAM's doctoral studies, and we have a DST-based paper. In past 2 section, and again, we have the first paper canvas from MAM's postdoctoral studies, and we have Malika Baduri and Kandimari et al.'s past 2 versions. And following to that, we have Hori Harmony web server papers. And now for the next week in Caps Lock, we have another set of interesting stories, and we have titled it Prevention to Protein Modeling. And please stay tuned. And we have 2 photos. I request ma'am to give some words about this.
Yeah, so these are all random clips, but some years ago, someone in the lab, I think Soni Malhotra, who is here in the middle, she proposed that we observe lab birthdays. And we started doing that. And these are pictures from our lab birthdays over a few years. And you can see there I am in the middle and my daughter is on one side. And by the way, this all used to be like a family affair. So we do observe, and our lab had many mothers, and you can see Harini holding her child too. And you can see the same sort of a lab birthday photo, but in a different year. Yeah, so these are some kind of random pictures that we wanted to share with you. Thanks. We have been answering the questions then and there. So we have dealt with some of the questions, and there has been one more. I'll just read out because of the fact that we are in the Q&A session. Have there been any improvements or updates in the structural alignment algorithms since the first version of PAS 2? All right. Gandhima, Uman, would you like to take it? The question is, have there been improvements in the PAS 2 algorithm and during the updates?
Gandhavati has already answered. Maybe she can
All right.
talk.
Has been. All right. Thank you.
Hello, ma'am.
Yeah.
So I actually, I mean, comparer is been there, and definitely comparer always performs, efficient alignment. But there are programs for the initial alignment, like we have used LSQman earlier, and there was certain, challenges using it on multi-members. Then we had, so there are over time, there are algorithms got developed, and we have also done this comparison of various programs. This was published in Advances in Computing. We have compared, I think, most of the structural alignment tools, right? Which one performs better? So over a time, like from 1998 onwards, there are like then and there programs keep getting developed, and it's being used by the scientific community.
Yes. Yeah. Thank you, GA. And thanks for the question. All right. So we will stay tuned, and we will see next week, same time, Thursday, 3 p.m. Until then, please take care. Bye for now.